Good afternoon, everybody, and welcome to this webinar on the IMI2 Core 10 topic, Biomanufacturing 2020, Development of Innovative High-Throughput Analytical Tools and Methods to Characterize Cell Culture Fluid During Development in Commercial Cell Culture Processes. My name is Catherine Brett, and I'm going to kick off today with a little introduction on how to use GoToWebinar. And after that, my colleague, Colin Carroll, who's the scientific officer responsible for this topic, is going to give you an introduction to the call and some of the procedures. One important thing to note here is that from this call on, we won't be using Sophia anymore for applications. We're going to move to the European Commission's Horizon 2020 tools, so watch out for that. After that, it's on to the main event of the afternoon, which is a presentation of the core topic by Luke Coopers of Genzyme. And then it will be plenty of time for questions and answers, and for that we also have Roland Wagner of Rentschler Biotech and Thomas Sentner of Sanofi on hand. So, how to use GoToWebinar? It's very simple. You want to listen, you can either use your computer's speakers or you can dial in. Just select the options as shown on the screen. And if you have any difficulties, obviously check your speakers are switched on and check your sound to make sure the system is picking up the right speakers. At the top right-hand corner of the screen, you should be able to see the control panel. You can expand and minimize it, and it's pretty self-explanatory. You can make it go full screen. You can see what your microphone status is, and very importantly, if you want to ask a question over the phone, you can raise and lower your hand to attract our attention. One very important point for a lot of you is going to be asking questions, and here you have two options. The simplest is to ask your question in writing by typing it into the questions panel and clicking on send. You can also ask a question over the phone, and for that you need to click on the raise hand icon, and we'll treat all the questions in one go at the end. Before we get started, just a couple of housekeeping notes. We are recording this webinar and we'll be publishing it online afterwards for people who couldn't make it today. We'll also be publishing the presentation slides, and we'll be circulating a participant list so that you can network with each other. And on that note, I would like to hand over to Colin Carroll.
Okay, good afternoon, everyone, and thank you, Catherine. My name is Colin Carroll. I'm a scientific manager here at IMI, and I'm managing this topic for the organization. So just a more detailed overview of what will be included in this webinar. As Catherine said, the main feature of the webinar will be the discussion of the topic. So all aspects of the call topic will be presented by Luke of Genzyme. The objectives, why we need a public-private partnership to achieve these objectives, structure of the project, expected contribution from applicants, and what the industry will also bring, and the key deliverables. We're not going to go into detail of the rules and procedures, which are obviously very important, but they're not for today. Instead, we're going to have another webinar in the new year, 9th of January at 2 p.m. Brussels time, where we go into detail of the rules and procedures, and we can answer any questions you may have on those as well at that time. There's a link there. It's on the same page as the registration for this particular webinar, but if you follow that link when you get the presentation, then you can register for that as well. So I'm going to start with an introduction to IMI, who we are, why we're formed, and what our objectives are. So we're a public-private partnership, the public part being the European Commission and the private part being mostly FPA, which is the European Federation of Pharmaceutical Industries and Associations. It's the umbrella organization for all the big pharma companies. And our aim is to facilitate open collaboration in research with the aim of advancing development of medicines for patients, for everybody, and with a focus especially in areas of unmet medical need. Those of you who are familiar with IMI will know that we've evolved from being focused very much on the early stages of drug discovery and research, and we're focusing much more towards the patient, delivering medicines for the patient. And that's where this topic fits in, is guaranteeing access to a good quality product every time you go to make some. So more specifically how we operate, so we fund multidisciplinary consortia to address challenging problems where one particular organization or industry cannot solve these problems by themselves. So by combining expertise and risk sharing in terms of funding, the idea is to build collaborations with a defined endpoint where the different the different expertises learn from the best practices from each other. So, for example, a collaboration between industry has got a certain way of working, SMEs have another way of working, academia works in a different way, but by combining the best practices of all three, you can really make a strong advance, and that's what we do. Overall, we offer ourselves as kind of a neutral platform where such work can take place, and also where perhaps conversations that are difficult normally to have can also take place as well. So to explain, I'm going to go a few slides here explaining our project life cycle. I think this is quite important, especially for those of you who are familiar with the Horizon 2020 or FP7 funding opportunities from the European Commission. There are key differences that we follow, and those are important that you know. Most of our rules are the same, but I think it's important that you do know what the differences are. And the first difference is where our topics come from. So I've got a slide here which has got the topic definition stage, and it says industry. So our topics come about by groups of interested industry organizations coming together in regular meetings and discussing what sort of challenges they face and what challenges they think that should come under the IMI umbrella, i.e. something that will where the industry can cooperate with each other, but we're also there's a public need, and therefore the public will benefit as well. And coming out of these industry discussions is a topic text, a draft topic text. This topic text is then consulted with all of the European member states to ensure that the public need, the public viewpoint is also addressed, and then it is launched as a call for proposals. This call 10 should be launched in the coming days. I think Catherine's already mentioned that we have published an updated version of the text on our website. So there is a good version of the text on the website, which will be more or less the final version. So if you haven't already looked at that, I would suggest you do so. So we're at the stage, once the call is launched, we're at the stage one of a two-stage process, and stage one is where the groups of applicants apply for the public funding half of the consortium only. So in the topic text, you'll see there's a clear industry budget and there's a clear public budget. What we're asking you to do is to form consortia and address the public side of the topic text and account for all of the public budget. So we have an example here on the screen of a typical consortium. We have, obviously, your consortium should be tailored to the actual topic in hand. It's important then that you submit your short proposal by a deadline, which is in March, early March. And this is then evaluated by an independent panel of evaluators. And the best proposal submitted then goes through the next stage. And this is a very important difference between us and Horizon 2020, is that we only ever fund one proposal from the short, we only ever select one proposal from stage one. So this is our main competitive step, and therefore it's essential that you get yourself into a consortium which is truly, truly excellent, because we only ever put one forward. So you have to work really hard at this, and it's going to be my main piece of advice to you, is to build a very strong consortium as early as possible. So following the evaluation, then the successful applicant consortium merges with the existing industry consortium through a formalized meeting which we organize in the office. But the concept here is not that you stay as two separate consortia. You're not an applicant consortium, an industry consortium. It's a true merger. So at this stage, we prefer you to think of yourselves as a single full proposal consortium, and this is the key element to ensuring you have a successful project, in that all partners are equal, everyone works together, everyone shares everything. And that is really important as well at this stage. This consortium also submits a proposal. It's a much more detailed proposal at this stage. It's got a very strong work plan consisting of milestones, deliverables, and so on. And this proposal is also evaluated by an independent panel of evaluators. And if all goes well, if it's recommended for funding, then we go through the grant preparation stage. The most important element here, I suppose, is the preparation of a consortium agreement, and this is a legal document between all the members of the consortium, and it's where things such as IP rights and governance and things like that are defined. So that's how we work. I think that, as I mentioned, the key differences are stage one, only one proposal goes through. So we work really hard at putting together a good consortium and writing a good proposal. I think that's the main step, the main point. So to submit a proposal, as Catherine said, those of you familiar with IMI will know that we've used a tool called Sophia up to date, an online tool where you submit your proposals. However, we have now changed and we are now using the Horizon 2020 tools of the European Commission. This should make your life a lot easier because there's a lot of standards there which you already, many of your organizations will already have. You can find your organization name there. Especially if you're familiar with the Horizon 2020 tool, our submission system is more or less exactly the same. There's a couple of specific IMI options you'll have to do, but they're very clearly laid out. My recommendation to you is that as soon as the call is launched in the Horizon 2020 tool, if you're interested in applying, go in there, have a look at it, play around a little bit to make sure you're familiar with how it all works, so that when it comes to finalizing your proposal, you're certain of everything you're doing and you're not doing a last-minute panic trying to get a piece of information which you're missing. So in the Horizon 2020 tool, there's two aspects to the proposal. The first aspect is kind of what we call Part A, which is administrative detail. Most of this you will find automatic when you look up your organization name in the portal. However, there's some detail required as well. But the bulk of the proposal, the main bit of work you'll have to do is to prepare a PDF document, Part B, which is your actual submission itself. One thing to note here is that we again differ from Horizon 2020 in that because stage one is our key competitive stage, we have increased the page limit of this to 30 pages. So in Horizon 2020 it's normally 15 pages. Here we have 30 pages, and we've also added some other aspects which are not normally in the stage one of a two-stage process, those being implementation and a list of participants. So in implementation, it's an outline of the work plan you're going to do, and then participants, it's a short detail of each of the organizations and key personnel who will be in your proposal. So this template is available on our website. It will also be available in the Horizon 2020 submission tool. And again, I recommend that as soon as the call is launched, you go in there, you download this, and you have a look to make sure you get all the information you need as soon as possible. Okay, so once the proposals are submitted, as I said, they will be evaluated by an independent panel of evaluators. This panel uses evaluation criteria to assess all the proposals received. Therefore, when you're putting your proposal together, it's very important you know what you're going to be judged by. And therefore, while I'm not going to go through all these on the screen here, they're here in the presentation for you to look at, but these are the evaluation criteria we use. There are three criteria: excellence, impact, and implementation. And as you can see here, there are sub-criteria in each of these main criteria. During the evaluation, we will go through each of these sub-criteria and we will look at your proposal against these, and we'll come up to an overall score. So make sure you do meet all of these criteria when you're putting your proposal together. And that's the one for quality and efficiency of the implementation as well. Okay, so just to finish off in a few remaining slides, these are tips for how to write a successful proposal, and this is based on our experience. This is call 10 of IMI 2. We had 11 calls in IMI 1. So these tips are based on a long experience of what works and what doesn't work. The first thing is, of course, read all the material you're given, especially the topic text. It's crucially important. There, at the bottom of the topic text, there's a section called call conditions. Make sure you read those as well. These are important rules you may have to follow for the specific call. I've said before, I'll say it again, begin forming your consortium early. This is probably one of the most important things you can do. And make sure you get all the expertise you needed to put together a viable proposal, because quite often IMI topics need you to reach out to people that you're not already in touch with. And if you need to do that, you make sure you do it quite early. Crucially important, make sure that the reviewers have all the information they need within your proposal to make their evaluation. They are requested not to look beyond the proposal that is submitted. So it's very important that you don't assume they will know something. Make sure you put it in your proposal itself. I would, so this next one, finalize and submit your proposal early. We do get it where, very often people wait to the last minute to submit their proposal. And while the Horizon 2022 can deal with the capacity of a lot of people submitting proposals at the last minute, if you have a problem, you will not have time to address that. So my recommendation is to submit your proposal early. You can always resubmit later. So I would test your submission early to make sure you can see that you've got everything correct. And one thing you can do is, if there's anything you're not certain about, please contact us. The email is info.desk@imi.europa.eu. It's important that you do contact us and not the industry topic writers, as we do not want to have any suspicion of conflicts of interest. A few common mistakes based on our experience. The first thing that happens to your proposal before it goes to evaluation is it has to go through a very quick admissibility and eligibility check. These are very straightforward, but still we get some people making mistakes. The first one is, if the submission deadline is missed, it will not even go for this check because we will not even receive the proposal. So make sure you submit on time. We still get emails from people saying, I didn't manage to submit on time, but there's nothing we can do about that. The second one is important. You have to have in your consortium at least 3 legal entities from at least 3 EU member states or Horizon 2020 associated countries. That's an eligibility criterion. If you don't have that, you will not go through to the evaluation. At the evaluation itself, we find that proposals that are not successful, perhaps they have not addressed all the objectives of the topic. So under IMI, it's important that you address everything in the topic text. You cannot just choose your favorite little piece and address that. While you will be evaluated, you will not be scored very highly if you do that. So make sure you address all the topics, and therefore it is necessary to have all the expertise to address all the topics. One thing that's become extremely important for us in IMI and in the European Commission as a whole is that all our projects have a strong impact. And therefore, if you have a proposal which you think is scientifically excellent, and could well be scientifically excellent, if you cannot prove that this proposal will have a strong impact as well, you will not be scored highly on the impact criteria, and you will likely not go through to the next stage. So again, please make sure you address all the criteria when you're putting your proposal together. One specificity of IMI is that we're a public-private partnership where you will have to complement the industry consortium, and therefore make sure you do address this in your proposal as well. Explain what you are doing and how you are interacting with the industry side of the consortium. So just a few tips on how to find project partners. Obviously, network with your own contacts. This is probably the key. You have to exploit your own network. Catherine said already that we will share the detail of the other webinar participants, those who agreed we could do so. So you will have an automatic list of other organizations interested in this particular topic. Definitely network with them. There are a number of partner search tools to facilitate you as well, and the list is there. We have a group called our States Representative Group. These are representatives from each member state and Horizon 2020 associated country as well. They are very, very familiar with IMI. Their job is to facilitate you to apply for IMI calls, so I would definitely get in touch with them. They're similar to the national contact points from the European Commission, and you can get in touch with those as well. And of course, network further as much as you can, social media, such as our LinkedIn group, and so on. One thing I would say is that at a recent meeting where we were discussing how to help partners find each other to put forward a good consortium, one person suggested that if you go into a consortium acting as if you are the coordinator, i.e. you're the one putting the consortium together, if you go in with that sort of attitude, you will find the people necessary to get a good consortium. And even if it happens that you are not going to be the coordinator, you are likely to find a good consortium with a good coordinator anyway. But you really need to have this kind of proactive attitude to go and finding people. It's probably not good enough just to rest with the people you already know. You have to go a little bit further than that. Okay, this is my last slide. It's about SME participation. So SMEs are extremely important for us at IMI. as a group of organizations who can really drive the results of the project through to having that impact on the patients. And therefore, we really want SMEs to get involved in all our topics. And I think especially in this case, there's a lot of activities within this topic where SMEs could and should be involved. So again, I would recommend that if you're putting together a consortium, think about what SMEs can really help you drive the results of the project through to the market, to make sure that the results are actually tangible and will be useful. So that's my last slide. I'll just reiterate that, you know, if you want any further questions on this, you can obviously ask them at the end of this webinar. But afterwards, please do contact us at infodesk@imi.europa.eu. And with that, I'm now going to hand over to Luke from Genzyme, who's going to give you the main presentation on the topic itself.
Okay, thank you very much, Colm. I have to make a small correction. I'm from Genzyme, but I'm also from Sanofi. So the company is Sanofi. We were acquired by Sanofi in 2011. So that's a small correction. So the title is Biomanufacturing 2020. So looking forward to 2020, how we get to biomanufacturing at those times. So it's about the development of innovative high throughput analytical tools and methods to characterize cell culture fluids during development and commercial cell culture processes. Can I get the first slide? Thank you. So the need for public-private collaboration. So this was a project which was coming from the ACTIP from the ACTIP organization. So ACTIP stands for Animal Cell Culture Technology Industrial Platform, and so that's a little bit the European Association of the biopharmaceutical industry, and we have about 45 members with the major biopharmaceutical companies in there. And while we were sitting together, we were feeling that there was still a lag, still a need, still a gap in the technology we are using. We all know that the animal cell technology is an established platform for biopharmaceutical production, so for production of biologicals. But we thought that it could be improved a lot, and there are still a lot of issues. And one of those is, of course, the cell. And there are still a lot of questions about cellular and systems biology, physiology, bioprocess science, which are relevant for animal cells. There's still a lot of knowledge to be built in that. The cell is still, I would say, a black box for us. And so we were thinking about that a more multidisciplinary approach was needed to get a better control and a higher prediction of the cellular behavior in the development, but also in the manufacturing, of course, of the biopharmaceuticals. So there's a poor understanding how bioprocesses impact cell and protein quality, and also there's a lack of easy-to-use, fast, predictive tools and methods. At least that was our opinion. And so that is really an ask for the development of innovative analytical technology, so that we could have faster, leaner, more controllable, cost-effective, in one word, more industrious production. Because if you compare the biopharmaceutical production with some other manufacturing, I would say, other manufacturers, like for cars or something like that, we still have a far way to go. So these novel methods to measure cell culture parameters and product quality attributes, they should lead into a more efficient production platform, and also a better process understanding, because we think that those are combined, so that when you have a better process understanding, you could have an increased efficiency of the animal production platform. Next slide, please. And also because, of course, those development of analytical tools is not really the core business of the biomanufacturing companies. We thought it was a good idea to reach out to the academic and small media enterprises, the SMEs, because we had a little bit of feeling that we know a lot, we can measure a lot into the upstream part of this, but we want to measure more. More means measuring more, means we have better control on what we do, and this also will have its effect on the critical quality attributes of the product. So we think that we still are working with some old, that there has been not too much innovation in all of this, and that is why we are reaching out to the community of the academia and SMEs to really get a grip on this and to really come with new systems, with new technologies, with new analytical testing, and we hope that there's also a little bit outside the box thinking in this, so that we can really bring innovation to this field as well. Next slide, please. So the overall objective is to develop a better understanding of the production cell, the cell itself, but also how the cells behave in its environment, and how this could be correlated to productivity and product quality. I think that is a very important objective in this. And for that, so we would like to enable novel, easy to use, fast, high throughput, call it whatever, analytical feedback control methods for parameters that can be measured, and maybe there are new parameters we're even not thinking about, can be measured during the development of biopharmaceuticals. So there are the high throughput methods we are thinking, so small testing volumes, miniaturization, automatic sample preparation, so that we can not only measure in the labs, because the labs are most of the time away from the manufacturing area, but that we can do this at line or even better online. And for those online, we are looking into novel methods or novel methodologies, where we look at the non-invasive testing, so like spectrophotometric methods and technologies that we can use to see how the quality of our cell culture is going, how the cell culture conditions are, and looking then in cell density, viability, amino acids, trace elements, vitamins, because, for example, the amino acids and the vitamins, we still have to take a sample, we have to go to the lab, we have to put it into an HPLC to measure those vitamins. So if you really want to take in situ samples, then that means that there's definitely a lag between the time that you're taking the sample and the time that you are measuring the values of, for instance, the amino acids. Next slide, please. The other thing is also, so feedback control, that is a little bit of process analytical technology, where we are looking at tests where we can say, okay, now we have this outcome of this test, so now we can adjust this or this parameter, so that we, in this way, we are in a much better control of the whole cell culture, and we can even guide the cell culture in the way that we want it. For that, of course, you need some algorithm or data analysis, so this is also part of what we were thinking of. And then coming to that data management, of course, if you do these online measurements and you measure, for instance, let's say every minute or every 5 minutes or even every half an hour, you have a lot of data, and we have to make sense of those data. So we are also looking into data management tools, but also in knowledge tools, because that is where it's all going. So we want to build more knowledge on the upstream cell culture to get a better control of our production and also of the critical quality attributes of our products. Next slide. Next slide, please. So there are still fundamental questions that need to be answered, as I mentioned earlier, in systems biology, physiology, a better control and a higher prediction. That prediction is quite important, that we know what the cell is doing. The cell is still, of course, a living organism, and but we know how to predict its behavior more and more, and that's what we learn by this project. it has a big impact. The production itself, and for instance, if you take the cell culture media, it has a big impact on the quality and the safety of the final product. So the critical quality attributes, so the characteristics of the protein, are playing an important role in this, and this is something that we really want to look at. How can we control these? So that means also that we are looking into the molecule itself, so molecule heterogeneity, where you talk about proteolytic processing, post-translational modification like glycosylation, isomerization, amino acid changes, oxidation. So we are looking in how to test this, and then I have to say also to test this as close as possible to the production itself, so that we do not have to run to the lab and then wait for a few hours or even a few days before we get all those answers. So the more we can measure and the more of those critical quality attributes of those proteins we can measure, the more we are in control of our culture. The same is true for process-related impurity. Think about host cell proteins, lipids, DNA, process reagents, media components, for instance, or process components. Most of you know that we use sometimes antifoams. We also use sometimes shear stress compounds like poloxamer, media additives. These are all process impurity, and we would have to have a better testing for all, and a more easy and a more rapid testing for all of these. So the expected impact. So it should lead to more effective products. Thank you. More effective products, more predictive, more economic, more competitive processes. This should lead to cost saving, but of course keeping in mind that we have to produce safe product. And for those cost saving, of course, it's our industry and the healthcare system. We all know that there's a lot of discussion out there now on the healthcare system about the cost of the biologicals, and this, of course, by having a better control, by doing some testing and respect for safety, that we can really work on this. The other thing is as well that we would have a qualitative and a consistent manufacturing. That means that we want to increase the supply chain reliability, and as such, by increasing that supply chain reliability, we would reduce drug shortages and definitely securing that delivering of these therapies to patients, which is of course the end goal in all of what we are doing. So this also brings the biopharmaceutical industry to a next level of competitiveness in the world. So as we can produce in a more efficient and more cost-effective way, and that we can increase the significance, but that's not only that, but we can also bring more products to the patient, which is which of course is also a goal of in all what we are doing. Next slide, please. So the suggested architecture of the project. So normally a project works with work packages, and we haven't defined any work packages at this moment, because I think and we thought that the work packages should be developed once we know the consortium, once we are building this, I would say, company and then academic SME consortium, where we draft that project. So we are still a little bit waiting what all the proposals are coming up, and based on all those proposals and based on the selection of the consortium, we will definitely define work packages. The duration of the project is expected to last 48 months, and the budget, the total budget is 9.4 million euros, meaning that 4.7 coming from the FPA companies, and 4.7 million is coming from the EU for the consortium to be built. Next slide, please. So what are the expected contribution of the applicants? We are looking that the applicants develop innovative, automated and highly sensitive measurement tools for whatever you call, but for protein, lipids, growth factors, amino acids, but also gases like CO2, O2, and some of those equipment already exist. Some of those tools exist, but we even want to look at the tools and maybe we can improve them, or maybe we can approach them by a different way so that we have a different type, maybe an easier type of measurement, maybe a more consistent measurement. So for lactate, glutamate, LDH, of course, also as much as possible to develop this in a high throughput manner, so that we can at the same time measure these parameters, but also measure product quality attributes, purity, aggregation, glycosylation. Some of these tests are online or could be done at the bioreactor. Some could be in the lab. But the closer to the bioreactor, I think, and the faster that we know the results, the better that we can control our culture. Innovative microscale fed batch cultures we are looking at. Development of rapid spectrophotometric methods for cell culture monitoring like Raman, near-infrared, FTIR, whatever. Maybe there are new technologies out there, and that is pretty much of interest for that, because we are really looking in innovative tools that we don't have on hand at this moment. Next slide, please. So also to develop process analytical technologies, as I mentioned earlier, because this is of course something that we want to do, so that we know with the designs of experiments, we know that a little bit where our culture is going, and that we can make minor changes and do not always have to go immediately to the regulatory to ask, can we do this or can't we? All this comes down to having this better control of the whole production, I would say, the whole manufacturing process, but especially all on the upstream, so the cell culture process. Looking in companies that are companies or academic groups, definitely, that are specialized in high throughput assays, specialized in all those omics. You have proteomics, metabolomics, glycomics. I think at this moment there are about 10 omics which could really be used in studying our culture, and from that studying of our culture we could then go and say, okay, this is and this and this and this what we can develop, so that we are in much better control of our culture. Also the management, the interpretation and the modeling of the complex data, so making sense of those data, I would say that is something that's very important. And then so companies that or academic groups that are specialized in development in online tools, online probes. I'm thinking also online probes that could be used, for instance, in disposable bioreactors, which are really something that is really coming up, augmenting in our world. So disposable cell culture bioreactors. Next slide then. So the industry consortium is composed of the following companies: the Sanofi and Genzyme, of course. Then you have GSK, you have UCB, Rentschler Biotech, Pfizer, Bayer, and Synton. Those are the partner in this, I would say, the industry consortium. Next slide. So what are expected contribution of this industry consortium? Of course, we have a lot of expertise. We are producing proteins, or we are producing biologicals, and not only proteins, but there are new, I would say, new forms coming up that can be produced as well. So you have high throughput, we all have it, biopharmaceutical cell line development, We already use robotics analytical testing. Quality by design is something that we use very often. Sample technologies, data handling tools. So that is something that we can contribute. Program management, of course. We have a lot of technical expertise and support. We also have a lot of scientific knowledge. And I think a big advantage in all of this is, let's say that you as an academic group or as an academic consortium or academic SME consortium have developed a new tool that we could really test this tool in real conditions, really qualify and validate these tools. I think that's a major advantage, which is not always present in academic groups nor in SMEs. And of course, we can also deliver resources. Next slide, please. So what is in it for you? So you, of course, will have that translation, that validation, that implementation at larger companies, companies that are producing, that have commercial processes. So for commercial purposes, of course, making partners strong in this highly competitive environment. So I would say this is also an opening, a gateway to those larger companies. So there's the connection and the collaboration with those leading companies in the biopharmaceutical industry. Companies, of course, that are experienced already in the production of proteins with animal cells. There's the conditional access to companies' infrastructure, and of course, what I mentioned earlier, the translation and validation of those new innovative technologies in real conditions. Next slide. So some of the key deliveries, this is a list, it's not limited, so that means that it's open. If you could come up with new, it's even better. But we were thinking about a deliverable high throughput testing, novel analytical methods, feedback control, measurements or testing that measure key attributes of the cell culture processes. Of course, you know that we are working in a GMP environment, so it's important that at least when we do this with the bioreactors, that these tests or these technologies are working in a GMP environment. Miniaturized microfluid-based technologies is something we were thinking of. Looking at high throughput product quality target product profiles, how to monitor that, how to control cell culture processes. Online, at-line, or even in-line tools for parameter measurements. New technologies, so PAT, coming back then, PAT, process analytical technologies, which is where that you can, by testing, you can really direct your culture and you can make small changes to your culture. Of course, all this, again, we are working in a GMP environment. We are looking for best suited instruments or tools to perform those analytical methods. Next slide. Tool to measure critical process parameters. Oxygen is, for instance, one. We know how to measure oxygens, but again, I come back to maybe there are other ways to measure oxygens. It's still measuring oxygens in those disposable bioreactors is even more difficult, so maybe come up with new technologies there. And it all should lead to that better process understanding. That is, in fact, we are looking for that better process understanding from where we can go then from this process understanding to a more predictive cell culture and a better control on the cell culture to produce those high quality product. We are also looking in data management tools, tools for the integration, making sense of data, and building also knowledge database and statistical tools so that you, for instance, can do trending, all this stuff. Some of those already exist, but we are looking at new ideas, new approaches, how to tackle this in another way, so that we have maybe more insight in how that our culture are performing. Next slide. And I think that was the last slide. So now I guess that this is open for questions.
It is indeed. So thank you very much, Luc, for your presentation. I'm just going to unmute Roland and Thomas so they can join in with the discussion in a moment. Just a reminder that today is very far from being your last chance to ask questions about this topic. If you have any questions, you can send them to us by email to info@imi.europa.eu. It's our job to help you, so no matter how small or how silly you think the question is, do get in touch. As I mentioned earlier, there are two ways to ask questions. The simplest one is by typing your question in writing and clicking on send. You can also ask a question over the phone, and if you want to do that, you can raise your hand. I think I can see that one person has raised their hand. Moment. Yes, Stephen Van Helden. Stephen, okay, I've unmuted you, Stephen. You can go ahead and ask your question.
Okay, thank you. Yeah, Steven van Helder from the Pivot Park Screening Center. You mentioned in the call text specifically looking for partners that are specialized in high throughput screening assays, HTS. Do you refer to HTS as used in early discovery in pharmaceutical industry? And if so, what kind of, well, what's the background of the wish to have them incorporated?
I think it's more HTS in more general way, so that you look at certain parameters and that you can measure on the one hand with a lot of samples maybe at the same time and measure the same parameters at the same time in a lot of cultures or a lot of samples coming from your culture. But also a very fast way of testing, because as I mentioned during my talk, I think that the time is playing a role in this. So the faster that we have all of this, let's say that you take 10 samples and you can test them at the same time for those parameters, and you have within a few minutes or you have your result. That means that you can really steer your culture by changing your parameters, for instance.
Oh, one comment from my side to all that listen to the webinar. When you look on the slide number 4 of the presentation, you see at the first project that the overall objective is to develop a better understanding of the production cell and its environment and how this is correlated to productivity and product quality. That means, although the process expresses measurements, methods, and tools as target, we have to emphasize that these technologies cannot be seen totally detached from a deep understanding of the cellular events and the cell environment interaction. Please consider this when you have your consortia, when you try to find your consortia and your colleagues to do this. Therefore, you find this addressed as the first point here in this objectives. What I have to say to that is, I think to keep it in mind is when we target a trustable process, we have to know that the basics for a robust and accepted high throughput analytical system is also addressed to intelligent cell line, which is high degree of transparency, for example. So that means that you have ideally a name that is therefore obtained by approaching from both sides in this project, the cell environmental system and the corresponding robust measurement methods. In this project, and I actually, one sentence, for example, or one comment on, in this project, and actually in all projects, it is expected to think that what Luc already said is outside the box. Yeah. Here you should design a science-based process space. Yeah. This is what the industry needs, which cannot be done, for instance, by also integrating disciplines that are far apart from each other. And so you can bring new ideas and concepts to prevent us, and that is what I think, us from becoming encrusted. That is what we had to do because we have the technologies that are 20 years ago, what Luc presented in this presentation. Okay?
Okay, thank you, Roland. We have a question that's come in through the questions panel from Ioannis Papantonio. Would a consortium focused on ATMPs be of interest for this call? Of course, applying HTS and QBD and PAT, however, for cell-based products.
If I may answer this, so ATMPs, advanced therapies, medicinal products, definitely, I guess that this could use some of the technologies which are developed in this project. But I have to say that the focus is not on ATMPs. The focus is more on the cells used as production vehicles of proteins. I have to say to this that there's also an ATMP program coming up, and I know FPA and other European organizations are very much interested in ATMPs, and we are looking into as well into how to improve manufacturability of ATMPs and talking about gene therapies and at the same time also cell therapies. But the focus of this project is on cell production of biologicals. That is the focus of this.
Okay, thank you. We have a question from Bichara Faraj. Maybe a naive question. Is manufacturing of immunotherapy products applicable to this call, or is it limited to biopharmaceuticals?
What is meant by immunotherapeutic? Because if we talk about ATMPs, like you have, for instance, the CAR T cells, I would say, that is more part of the ATMPs. That doesn't mean that some of the technologies developed in this can be used in that field as well, but that is not the major focus. If you talk about those new products and combined products, the ADCs, so the antibody-derived conjugate, where you have some molecules attached to your antibodies, this is definitely in focus.
Okay, thank you. Damien Marshall has raised his hand, so I'm just going to unmute him. So Damien, I've just unmuted you. You can go ahead and ask your question. Damien Marshall from Catapult in UK. Doesn't seem to be there. Okay, if you have a question, then you can also send it in writing. We don't have any other questions for now. So I will just remind you that the call hasn't been launched yet, but the indicative topic text is available online. And you can download it and you'll have all the details there. And we really would advise you to read it carefully, and if you have any questions, come back to us. Okay, so that was an accidental hand raising there, so no worries about that. Okay, so I'm just going to unmute Damien. 1 second. Okay, Damien, I've unmuted you now, so go ahead. Sorry about this.
Hi there. Can you hear me now?
Yes, we can.
Excellent. It was just really to follow on from the questions that came previously about the ATMPs. Under the expected impact, it actually says that an important part of this is to develop tools that will be immediately fit for purpose for advanced products.
Wait, Damien, sorry, I'm going to have to interrupt you. The sound quality is very bad. Can you maybe send your question in writing? Because it's quite hard to hear what you're saying. Sorry about that.
Yeah, that's no problem. I can do that.
Yeah, okay. So while he works on his question, again, the topic text is available online. If you keep an eye on the IMI website, you will see when we do launch the call, which hopefully will be in the next few days or so. And we really would advise you to read all the materials, all the call documents very carefully. And if you have any questions, especially with this new application procedure, do get in touch with us. As Colm said, we'll have a whole webinar on nothing but our rules and procedures in January, and you're very welcome to come along to that. And again, that one will also be recording it and putting it online. I don't know if there's anything you want to add, Colm, on the call. We have the question. Thank you, Damien. Under the impact section, it states that technology should be fit for purpose for ATMP. How is this expected to be addressed within the consortia?
Coming back to my first answer is, so the focus is not ATMP. It should be written, or it should be read like this, that some of the technologies or some of the ideas or some of the testing that are developed in this project could also be used within the ATMP field. The focus of this project is the use of the animal cell for the production of biologicals, conjugates, or monoclonal antibodies or proteins. Again, there is probably something coming up on ATMPs within the next year or the year after that, which could be related to this project, but the focus is not ATMP. So the idea behind, and that is the phrase, and maybe it should be rephrased a little bit to give the right idea, is that the ATMPs can benefit from this, but are not the primary focus of this. The primary focus is the use of cells for the production of biologicals.
Thank you. We don't have any more questions or raised hands for the moment, so if you have a question, and I would urge you to type quickly. I think Colm would like to just make a little comment.
Okay, one thing, just to follow on from what Luke said during his presentation on the architecture of the project. Luke said that in the topic text they haven't given a structure for you to follow, and that will be decided at stage 2 in collaboration with the industry partners. I should just point out that at stage 1 you are expected to give an outline of how you think the work plan will be structured. Now, obviously, this work plan can then change for stage 2, but for stage 1, the expert evaluators are going to want to see what your idea of this would be. So please give at least an outline of what you propose, such that the project would look like, the structure of the project would look like.
Okay. We don't seem to have any other questions, so I think on that note we can say thank you very much to Luke for his presentation, and for Roland and Thomas for their input on the questions. Thank you to all of you for coming along. Thank you to Colm for your presentation, and I wish you all very good luck with your presentation, with your proposals, and have a good weekend. Goodbye, everybody.
Thank you. Goodbye.
Thank you very much.